In this video, we're going to start talking about how we take DNA and do sequence alignments and build trees from them. As we can understand that in biology, we know that the DNA is some sort of record of information, and that over the period of geological time, there has been changes along the way due to problems in replication because there is an inherent error-proneness to that process. So we're going to see how we use DNA sequences and understand how we can draw relatedness between different organisms. So we're not going to go into great detail about the various algorithms for how alignments are done or what types of alignment algorithms there are. We'll just use one standard one for multiple sequences. And we're also not going to talk about the theory behind tree building so much, as long as we understand that it is a process for uh, creating branches of relatedness between sequences. And there are many different algorithms and reasons why you would do such a thing, right? So first, just going to talk about things that are non-computationally expensive in nature in terms of looking at differences and likenesses of various things, right? So in order to do that, I first want to bring about a whiteboard, right? So we can draw on this. So here's our whiteboard, and let us change the page styling a little. Um, so let's just make this a little bit more custom in terms of how it will map to our screen, right? We'll do sort of a, that should be good enough, right? Anyway, my screen resolution is higher than that, but that approximates an HD screen. And so what we can do is think about how we are going to, just draw a bunch of letters around here. Uh, right. And so let's make that a little bit neater. And so what we can do is think about our letters here. And let's use a different pen to actually talk about looking at where these letters lie on this field. With that understanding, we can ask, what are the ones that are closest together? So I can see that A and B are close, as well as G and H are fairly close, right? And then we can think about ones that are close after that. I would say that these are equally as close to F. Um, and then possibly we have C and D, and then we have E. We can say it's all by itself mostly, right? And if we were going to draw some sort of relationship, we can think about how A and B are next to each other. And we also know that subsequent to that, we can draw like another sort of grouping. I would say that C and D might be closer to this cluster over here. So we can write C, D, which we know are close to each other, then F, then G and H, right? And then we can have E all by itself, right? Just roughly speaking. You don't necessarily have to agree with me, but we can get a ruler out and start to do this more mathematically. And so those things are close, and then we know that these guys are close, right? And then we can think about how C and D are close to this, and we can just draw another sort of linkage here, right? And then now, like, I ran out of space, but we can go like this, right? So what we've done here is called agglomeration. Maybe I spelled that incorrectly. And that was fairly simple. It didn't require any mathematics. It was just eyeballing these relationships. And as I said, we can also take a ruler and measure these distances, and we can actually generate something very similar to what we have here. By agglomerating them together. And so you can see that happened very easily. It didn't require a whole lot of math, didn't require a lot of computational time to do this activity. So going back here, we can think about, and that's one type of tree. We can call it a dendrogram, and that didn't really imply anything about evolution because there was no evolutionary information about it. It was just simply about distances, and distances are very useful in that respect because, like we can say, it's easy to see how far away things are. Now, when we talk about alignments, alignments are going to give us a score. And we're not going to discuss that in great detail because this is not strictly for bioinformaticians. This is more in general as a concept of how these things are working. And if you really need to know greater detail, then we can discuss that at a different time. Right? So what we're going to do now is try to, and there is a video here on how to do this in Eugene, and we're going to work through this example, but we're also going to go about finding the sequences that are here. And so basically, what I'm going to do now is I do have some of these things in my file system that are present for me, and I can bring those up very briefly. And we're talking about essentially the great apes, right? So essentially, I have a handful of great apes, and we should think about what the great apes are, right? So there is a definition of great apes. First of all, apes is a superfamily in our categorization system using taxonomy, right? So remember domains, kingdom, phylum, class, order, family, genus, species. And there's something called a superfamily. Well, first of all, primates are in the class of being mammals, right? So remember, class, order, family. So not that I'm a great taxonomist or anything like that, but I would assume that our order is the primates. And so primates involve things such as monkeys, apes, and also the lemurs, right? The lemurs being something that had been isolated to the island of Madagascar due to its separation from the African continent. And so we have those types of categorizations. And then amongst the apes, right, there is this superfamily of, well, it is the superfamily. And then we have the family called apes. And we also have the great apes and we have the lesser apes, right? What's the difference between a great ape and a lesser ape is you know, you would have to see the physical description as well as the genetic information. So what we're going to do now is go and look at some genetic information. And we're not interested in looking at all the genes or anything like that. We're using what's called some sort of barcode that differentiates these organisms from each other, where we would see that every single organism, individual within a genus or species would have the same sequences that differentiate them from another genus or species, right? So what we're going to do is first, let's look at a list of our apes, right? So as I said, we have this family of great apes, right? So we have in the genus Pongo, and we lost some of this sort of indentation, so I'm just going to do this like that. So we can see that we have in the genus Pongo, the orangutans, we have the Bornean, the Sumatran, and the Tapanuli orangutan. And so you can see here that there is some sort of species name associated with these. This is a text editor, so there's no formatting involved. So we do not see these italicized or underlined as they should be, right? In Word, you better italicize it or underline it because that is appropriate. But if it's plain text, you don't have that option. So those are the great apes. And notice that we humans are Homo sapiens here. And there are other types of humans that no longer are in existence, right? We can see here we have a handful of, a broader handful of these, right? Sort of in these multiple genera that we have here. So I'm just organizing it a little better so we can see that we have, stop, a handful of genera that are descriptive of or emblematic of our gibbons. So we have 1, 2, 3, 4 gibbons, and so those are the apes, right? The superfamily apes. We have the great apes and the lesser apes. So we can go and try to find out some information about that now, right? We have, let's say I have a bunch of two representatives of the Hylobates. So let's try to find a Hulak, right? A genus Hulak. So I'll just copy that. And what we'll do is go over to our browser, and we'll go to NCBI Nucleotide, right? So what we're going to do for this barcode is we're going to take something from the mitochondrial genome, or maybe we'll even look for the mitochondrial genome in its entirety, right? So we're going to go Hulak. Hey, look at that. It's one of the searches that are in existence, and I've never done this before. So let's try the Hulak mitochondrion. Right, so let's search that. And on the side, why don't we look up what one of these organisms look like? It's the Hulak gibbon. And we'll look at some pictures. And, well, there's one there. Right, so you can see what these gibbons look like very briefly. You can tell they are apes because they are obviously a primate of some sort. And not only that, they are lacking a tail, which is a hallmark of an ape, right? So like humans and orangutans and gorillas, they do not have an external tail, and that's one of the hallmarks of these types of primates, right? And then, so we got that. Hey, and we have these complete genomes. We have two of them. I don't know what's different about them, but we'll just go and download this one. So we'll just open this one up. They're different isolates, but they seem to be the same isolate, so I don't understand why there's two. And so what we'll do is we'll get the GenBank file once it's finished loading. And as that's loading, we can go back and consult our thing. So we have a Hulak. Let us try a, let's see, Symphalangus, right? Symphalangus. Let's try to find that. And we don't need to do this exhaustively. We're just trying to, you know, sort of get some sense of these organisms. So we're saving this file. And while that's doing it, we will search for the Symphalangus gibbon just to take a look at what it looks like, right? And yeah, so here we go. Come on. As it loads, we shall see this is a Symphalangus, and it is another type of gibbon, as you can see. And it looks different than the other one, but it also looks very similar in features. Once again, it is obviously some form of a primate that has a lack of an external tail, of a certain type of stockiness and musculature that is obvious as well, that we see that is common to the apes, right? So, ooh, something happened here, so let's redo that. And hopefully it loads soon. Yeah, so let's go try to download that again. And I think there was another one that we could be looking at just for kicks. Symphalangus. I know I have Hylobates, Hulak, Symphalangus, and let's look at the Namascus genus, right? So we'll just copy that and hopefully open up this. Still not saving my file. So let's go look at what one of these guys look like, this genus of gibbons. And there's the bad gateway thing again, so that's a nuisance. And so these are the Namascus ones. Let's go click on that. And at some point, we're going to try to get a sequence. If not, we'll just go with the FASTA. It doesn't matter. Here, let's just go click on the FASTA and let's see what happens. In any case, yeah, and so here are the Namascus gibbons, as we can see, images of, right? Also, once again, very standard looking primate of some sort. I would not be able to tell these guys apart necessarily because I don't know their names and what their what their definitions really are that differentiate them. Uh, and it's taking a very long time. But in the meantime, we can go to NCBI Nucleotide, and we can start to look for a symphalangus. Uh, yeah, so here's this. So let's see if we can save this. Otherwise, we're just going to do it the old-fashioned way of copy and pasting. Yeah, it seems to be thinking very hard. But you can see how long this sequence is, and that might be the reason why. It's probably something around the order of 17,000 bases, because that's approximately the size of a human mitochondrial genome. Yeah, so there's some weird NCBI issue that's going on. So in any case, we'll just do it the old-fashioned way with the FASTA sequence. That'll probably be the easiest way to do that. So it's done loading. Once again, all we got to do is go like this. Just copy and paste it into a text editor, not a word processor, right? So here we go. And this is FASTA, so there's no numbering and there's no double // And once again, we'll just open up our text editor, open up a new document, paste that in there. And here it is. And one great thing about this is I might want to actually put this at the end instead of where it is, because that'll make things easier, as we'll see later for some other things. And we shall save this in our folder. Right, so I'm going to put this in my Apes folder, and this is going to be called hulak.gb. Right, so there we go. So once again, it's simply a text file, and I realize it's not easy to see right now, but it is fantastically okay the way that it is there. Let's see. So we got that out of the way. Now let's do the symphalangus, right? So I don't know how to spell that, so there we go. Hey, look at that, mitochondrion. See, this seems to be a common thing that I did not do, so that's fine. Oh, I think it's because I had this checked off maybe. But anyway. So here it is, a complete genome. Now, remember, when you have a complete genome, there might be multiple isolates that are listed because not every individual is identical, right? And what we're trying to say is, and I'm going to prophylactically go with the FASTA. What I'm trying to say is that there are identicalness between genes, but then there are portions that are not genes that are prone to more mutations that we don't use in our analysis. But here we're just going to go drag this one open, and we're going to go back to this one and put in our Nomascus one. Let's see. Nomascus. Sure, we'll use this one. I don't remember how many species were in this genus, but there we go. And here we are, the symphalangus mitochondrion. I'm going to do the copying and pasting because I know there may be an issue. Remember, in science, there's always multiple ways to do the same thing, and it's important for us to be flexible enough to adapt to these types of challenges. So if I can't press a button to download the file, I can do it a different way, as long as I know what the file should look like, right? So I'm just going to paste it over this one, but I'm going to save it under a different name, of course, and I'm going to put this accession number at the end just for making my life a little easier. So we're just going to call it symphalangus, even though there are multiple ones. So we're going to save as. There we go, symphalangus. And that's our representative. And then let's go back to the browser. And as we go back to the browser, we can go back and see the Nomascus leucogenus. And look, there's a complete genome, right? And so we'll just go to the FASTA because that'll be better for us. So here we go. FASTA, waiting for it to load. And the thing is, you could actually play a game where you can like get a picture of each one of these and put them up against each other and try to see their similarities in physicality. But the thing is, physical aspects that you see while they are mapping to genetic information, you can have many things that look the same that are not related because of this concept of convergence, right? But what we're doing is looking at divergence from common ancestry in our DNA. So here is our Damascus leucogenesis, and I'm just going to copy this way because NCBI doesn't seem to be cooperating today. And here we are. And I'm just putting that there because it makes it easier for us. And this is a Damascus, so I will save this file as that. Right, so if we look at this, we have Hulak, we have 2 Hylobates, we have a chimp, a common chimp, a some sort of orangutan, another one of those guys. I'm gonna go pick up another orangutan, right? And because it's important for us to do this, so let us go and pick up another pongo. Let's do the pongo belly. And we're gonna close these guys off. And these are all partial, and here's a complete one. So we'll just go with the complete one. Once again, we'll just try the FASTA. That'll make it easier. And in the meantime, while that's loading, oh, it already finished loading. I'm going to do another search in this tab just for the hell of it. So I'm going to look up another type of ape that's extinct, and it's called the Neanderthal. Right? Homo sapiens neanderthalis, neanderthalensis. So let's go get that. And here's our FASTA sequence. So I'm just going to pull this down here. Okay, so we have these genes here, or this genome. It's the entirety of the mitochondrion, right? And once again, I'm going to just reorganize it a little bit just for my benefit. And this is abele, so P abele. Save as. Pongo. So we're just, oh, you know what? I keep on saving these as GenBank files, and they're not GenBank files. That's okay, right, for now. We just might have an issue later with that. But I think Eugene will be able to figure it out. If not, we just rename the extensions. So going back to the last thing was we were picking up this Homo sapien neanderthalensis. And now this is something interesting. Now look, it says that there's a reference genome. And this is very important for us to understand. Like I said before, not every human has identical mitochondrial genomes, right? There are differences in various locations of it. So when we have Neanderthals are also, an extinct form of humans that some of us have genes from because our ancestors had intermingled with them. Especially if you are from not from sub-Saharan Africa. If you're sub-Saharan African descent 100%, then you will have 0 Neanderthal because your ancestors never came in contact with these humans. And so if you're European or Asian, East Asian, especially if you're East Asian, you'll usually have a higher degree of this Neanderthal. And there's another extinct human, the Denisovan, which we will see is also something that if you are Micronesian, then you probably have a high percentage. And by high, I mean greater than 1%, right? So like 4% or something like that. Anyway, you can see there's many of them here, right? So what we can do is just take one of these, but you can see there's different isolates. This one's been very not, doesn't really have any further annotations to the name. And you can see there's multiple ones of these. So we can go to this one, or we can just go to one of these. And I'm just going to go to this one because I'm being lazy about it, right? At the end of the day, you want to probably be, depending on the questions that you're answering, you should be more specific about things. But for demonstration purposes, we are perfectly okay to do this, right? Just to remind you that this is not every single Neanderthal genome out there. This is one of the handful that we know of, right? So once again, we'll paste that here just to avoid NCBI failing on us. So I'm just going to get rid of that whole Homo sapiens thing and just write Neanderthal. That'll make it a little bit easier for us. One of the many. Right. Time to start labeling them properly. And in the background, I am going to rename these properly just to avoid problems. And that will make it a little bit easier when we start to do some of these analyses. Symphalangus is also one. Halobates, Hulak. Okay, so now we have a handful of these there. We're pretty good with this. We got all these guys and, at least one representative from each. But we're not complete yet. Whenever you're doing some sort of analysis, it's always good to have some sort of negative control, right? So a negative control that we would use here is something that we know is not a great ape, right? And that tends to help align these sequences better. So let's just pick some other primates. And, why don't we pick two? We'll pick a lemur and a monkey, right? So let's do pygmy marmoset mitochondrion. Let's see if that comes up. And the GenBank file you will find is better because they're going to be much more informative of where the genes are, right? So if we went to the GenBank file, we'll just do that. Right. Let's see if it's going to cooperate with us again and give us the ability. And then let us try a ring-tailed lemur, right? So here's another, oops, I should have been more specific. So it's called lemur kata, so mitochondrion. Those of you who recall an individual called Zubumafu or Zabu, whatever was named from that show, that's what this is, right? And so, once again, I'm going to go to GenBank just in case. And then over here, it's still loading. Let's see if I can save this. I really would like to have the GenBank file for this. And you know what? The easy thing for me to do is just copy and paste too. It's just like a lot longer. So why don't we just go to here, the GenBank file. We'll start there and copy and paste just in case it fails. Right? We're just expecting it to fail. So there we go. And that will solve that problem that we've been having. Right? So let's make a new one of that. And so, yes, much of what we're doing is tedious, but that's the nature of it. And at the end of the day, these things are present in a database for us, which means some scientists have gone about discovering these things, right? So let us save this and remember that this is our lemur kata GenBank file. And let's see if our last thing is ready. Bad gateway, exactly. So we'll go back to the other way of doing it, which is just copying and pasting. And this just goes to emphasize the significance of plain text, right? Plain text is not large, and it's a little bit flexible in the sense that you can easily parse it in a text editor. So here we go. And this individual here is the Calithrix, right? Uh, Save as. Here. And as you can see, it has the common name that I called it by. You can see that under the organism heading, it also tells us that it's a eukaryote, it's a metazoan, which means it's an animal. It's a chordate, right, which the subphylum is vertebrata, and you can, which is right here, and you can see that it is a mammal, and the subdivision of mammals is eutherian, right, which is something a little bit that we don't talk about. We don't talk about these categorizations so much unless you're a paleobiologist maybe or a primatologist. It's a primate, and it's some sort of monkey which has some sort of family here, and I've lost it by now. So there are probably many families or subfamilies that exist for this. In any case, we have our representative sequences, right? So let us go back and think about how we're going to do this sequence alignment, right? We're just going to go stop the recording now and pick up again in actually how we use these sequences.